This webinar today is all about exotic leaf miners. It's an update on a Hort Innovation funded research project for control, eradication and preparedness for vegetable leaf miner.
Well, thank you. Just following on from Aaliyah, Peter, Rohan and I will be talking about some biological and chemical control issues with the leaf miners. So the three questions we need to address: how are leaf miners managed overseas? What beneficials exist in Australia? But how will IPM strategies look like in horticultural industries? My part revolves around biological control. We had a project where we did an extensive review of overseas and Australian literature. And we also did a deal of field surveillance for parasitoids. As we've heard from other speakers, the problems with these leaf miners are universally associated with destruction of parasitoids by excessive use of non-selective insecticides and development of insecticide resistance within fly populations. A couple of shots from Indonesia, where I did a project on Leafminers in the early 2000s with ACR. To avoid chemical mismanagement, we need to learn from the overseas experience. That's the advantage of Australia. We often get these pests a long time after other people, so we can afford to learn from other people. So once we understand the role of the parasitoids, we've learned we have to monitor the pest activity to prevent economic thresholds. And also we need to avoid broad spectrum insecticides. So effective control depends on maximising the impact of natural enemies, but around the world, in most countries at least, the endemic parasitoid species have been found to been able to exploit the invading Leafminer species, and that's important. And also the non-crop hosts have played a role as reservoirs of these parasitoids. Just on these, before I go any further, I'd just acknowledge Aaliyah's fantastic work on taking images of a lot of these parasitoids, and also her magnificent artwork for the Hemitacinus, another parasitoid. So you'll see these through the talk. Leaf miners, the parasitoid wasps, have basically two major life history strategies, and it's important to understand the difference, I think. Eobionts paralyze their host immediately, so that fixes the size of the food available for their offspring. And once the paralyzed larva, they'll either feed on that larva, and that helps their egg development, because a lot of these idiobiont wasps, when they emerge, have virtually no mature eggs available, so they need to feed on protein and lipids from their host, from their target, to help egg development. Or they'll lay an egg either beside the larva or in the larva, and that will hatch and begin development. So they have an immediate effect with an idiobiont. The coinobionts don't paralyze their host, but they oviposit into the egg, into the larva. So the fly larva keeps on doing its thing until it actually pupates. So in the case of vegetable leaf miner, the larva leaves the leaf, it pupates in the soil, and it's that stage that the parasitoid develops in the pupa. So it's got a known large size food reserve. So they don't have any effect on the immediate population of leaf miners, but they will generate parasitoids for the second generation. So with the idiobionts, you can see particularly ectoparasitoids, wasps laying the egg in or beside the larva. It feeds on this paralyzed larva, which rapidly becomes dead, of course, once it's being fed. It pupates within the leaf mine and will emerge from the leaf mine. The coinobionts lay the egg in the larva. The larva then leaves the leaf, goes to the soil, and that's when the parasitoid starts to develop. The big message with these very small wasps, and they are very small, even though they're very beautiful, all these leaf mining wasps are generalists. They'll attack very many leaf miner species. And in fact, there are wasps. There's one we found for the first time in Australia, Neochrysochrys formosa. That's known also to be a very important parasitoid of a lepidopteran leaf miner, Tuta absoluta, which is causing a lot of problems in Europe and Asia, as well as South America. So this is an example where the leaf miner is actually, the parasitoid is actually targeting a leaf mine rather than a specific target target host species. So this is makes it very difficult, of course, to bring in a parasitoid such as that from overseas, because you've got major issues getting overcoming a host specificity situation. We found in the survey literature there's very large numbers of species recorded from Leafminer sativa. We've listed 71. You always worry about the extent of how well people have identified species, but the message really is that it's a very large number, and we had similar numbers with Leafminer trifoli and also Leafminer huidobrensis. A lot of overlap, but also some differences. Some of these species are only found in South America, some are found only in Europe, some are only found in China, but there's obviously overlap as well. Most of probably the family Elophidae is the dominant group of parasitoids, but there are a lot of parasitoids from the Braconidae, Pteromaelidae, and a lot of eucoilines. So Diglyphus, Hemitarsinus, Opius. One of the things about these names, they're often a lot longer than the size of the wasp, which is just the nature of taxonomy. So what is the situation in Australia? So what parasitoid communities already exist in Australia? And do we have some of these important parasitoids from overseas? Which really leads to the question, do we need to import parasitoids into Australia? Simple answer, Australia's got a large number of parasitoids which already attack Leafminer and also a range of other agromysids. I think we could found at least 50 species have been found to attack agromysids in Australia. And still likely to be a dramatic underestimate due to the lack of sampling. So we've got Hemitarsinus, Diglyphus, and Opius. But the important question really is, are these parasitoids really going to attack vegetable leaf miner, Leafminer sativa? And so to do that, we had to do a lot of surveying. And the only place we could do it, of course, was in the place where Leafminer sativa was. So Aaliyah and colleagues made 4 trips to the cesar and Torres Strait between 2017 and 19. And one of the tasks they did when they're up there was to collect leaf miner larvae and rear out parasitoids. And because of the quarantine nature of the place, that had to be done up there. So that was quite a tricky job, but it was very, very worthwhile. What was interesting was that we found at least 6 species up there. Zagramosoma, we can see over here is a beautiful striped red and yellow and black striped wasp. Hemitarsinus up here with its branched antler, they were present. Zagramosoma was the most common one. Parasitism rates were very high, up to 80%, and that we believe they were very important in slowing the spread of Leafminer sativa. So the question, of course, is why were the wasps there? Probably the most common agromysid that they found in the Torres Strait was Ophiomyia elisa-carpi, and that's a leaf miner that was first described in Fiji. It's known from India, and up until now it was known from Darwin, but it's widespread in the far north Queensland. So it seems to be a very widespread in the tropical areas. And all parasitoids that were found on sativa were reared out of elisa-carpi as well. And as well as that, there were 3 more species of native agromysids that were found between cesar and Cairns, which were also parasitized with most of these wasps. What was interesting was that wasps such as Zagramosoma and in fact Hemitarsinus were found at the tip of Cape York, but we find them in Victoria as well. Aaliyah was, in one of her other projects, was delighted to find Zagramosoma happily parasitizing a grass leaf miner outside of Condolblin in the middle of a January heat wave. It was 45 degrees when she saw that. So it's a widely adapted species. Zagramosoma is a species found in Indonesia and also described from Australia. So whether it's an Australian or not, we don't know. As part of the project, we had a master's student, Marianne Cocchio, who looked at leaf miner and parasitoid communities within 4 common weed species around Melbourne. She was interested in Leafminer brassicae on brassicaceous weeds. She looked at Leafminer kenapodi on Stellaria media, Phytomyza plantagenus on plantain, and Phytomyza syngenesica on on sow thistle. So you had 6 sites, great variation between sites. So just as an example, Leafminers Paris along the bottom line, these are months along here. It was most abundant in, this is just in quantitative terms, it was most abundant in autumn. The key message here at this site was there were 10 parasitoid species, 7 Eulophus and 3 braconids. Similar things were found at other sites. The 3 species that are highlighted in pink were species that were also found up in the Torres Strait. So the message really is we have plenty of promising biological control options in Australia. We certainly don't have any need to import. The all important thing is to protect what we have. And that's how we have to go back to these foundations of IPM approaches overseas, understanding the role of parasitoids. We'll have to monitor pest activity. Presence of mines doesn't necessarily mean you have to spray, and that's when the one mine doesn't mean to say there's going to be economic damage. Sampling's important, and activity might have to be done by monitoring for pupae or adults. This is monitoring now that once a pest is actually established. But I think it's really important to know that these sampling strategies and economic thresholds will be crop specific and must be tailored to Australian cropping situations if and when the pests appear. So in terms of avoiding broad spectrum insecticides, which is the all important third part, Rohan Burgess from Plant Health Australia will be talking about that for the rest of the session.
Yeah, so I'm just going to give a bit of a run over the project's work towards some of the chemical control options we've been reviewing and looking at. And as Peter said, the key sort of theme to the whole thing has been just trying to keep, the beneficials going. So when selecting chemicals, the things we're sort of interested in, first off, is just being aware that there's resistance overseas to a lot of these chemical groups, especially some of the broad spectrum things like organophosphates and what have you. And one of the things to note is trifoliate seems to have more resistance popping up than some of the others. And really, all this means is it isn't as simple as just sort of buying the cheapest possible chemical that's broad spectrum and throwing it on, hoping for the best. You've got to be quite selective in what you're doing. So I just thought I'd have a quick bit about the types of chemicals, I suppose. So broadly speaking, you can sort of think of chemicals in 3 different types. There's sort of a bit of crossover in that, but essentially you have contact pesticides, systemics and translaminars. Because the leaf mine larva is developing inside the leaf, you want something that can penetrate into the leaf to kill the larva rather than just a contact that's going to take out the adults. And that's why we've also sort of used that lens, I suppose, to look at what chemical options are out there. So next slide. As Peter noted, the beneficials are very good at controlling and keeping the leaf miner populations in check by themselves. So we really want to be a bit selective about the pesticides we're using and not take a sledgehammer approach to things. Cesar conducted a review as part of this project looking at the, I suppose, the toxicity disruption of most of these chemicals against, of commonly used pesticides overseas against the key beneficials for the leaf miners. And they've just sort of come up with this bit of a spectrum, I suppose, where you'll notice that things like organophosphates are more disruptive than some of the other chemicals like chlorantraniliprole and chromazine, stuff like that. So again, we've sort of used this lens of looking at how can we conserve the beneficials where we can going forwards. So that's sort of how we've gone about selecting some of the pesticide options. But there's also the question of what do you do should leaf miner arrive in the country, well, in a production area because it's already in North Queensland. How do we go about managing pests that are already here? because obviously, you know, farmers have a lot of pests to deal with. So I suppose the thing to bear in mind going forwards, if Leafminer establishes in your area and you're a farmer, is you want to be selective about chemicals regardless of what you're controlling, to try and conserve beneficials. And generally speaking, the horticulture industry is quite good at doing that anyway. So just as a bit of an example, in this diagram, if we're trying to control, say, whitefly, for example, which is in, I don't know if the cursor's working. If we're trying to control whitefly, for example, you'd probably be looking at more pymetrozine or spirotetramat as better options than imidacloprid, for example, just because it's less damaging on the beneficials for Leafminer. That sort of stops the problem of Leafminer becoming sort of something that was there in low levels with the beneficials in place becoming a problem. So I suppose the take-home message is, once Leafminer becomes established in the south, in the production areas, is just, we'll have to be a bit careful about what chemicals we use, and develop plans accordingly. Right, so the next slide's basically, this is how we've gone about selecting chemicals, I suppose, that look to be most promising for Leafminer control going forwards. As I said, we've sort of looked at it from the lens of their efficacy against, their toxicity towards the beneficials, and we tried to pick things that are less damaging to the beneficials. You'll note in this table we've got efficacy against different life stages of the Leafminer. So that's another thing that we'll have to sort of bear in mind. So some chemicals might be, like trimetrozine, for example, isn't very effective against the adults, but it's quite effective against the larvae. So it sort of won't have that immediate effect, I suppose, in terms of, you know, you'll still see adults kicking around for a little while or whatever, but over time it will be quite effective. The other thing in this table is we've looked at sort of a bit of a cross-check to see what chemicals, so all of these chemicals are used overseas for Leafminer control in various countries and such. And we've also looked to see which ones are used widely in Australia. And we've tried to sort of cherry pick a bit. And that's how we've come up with this list here, which is probably the more interesting to the growers in the room, which is essentially, through the project, we've developed pesticides covering 5 active ingredients: abamectin, clomazine, emamectin, spinosaurum, and spirotetramat. And if anyone's interested in finding out more information about them, the permits are publicly available on the APVMA permit database. But the take-home message, I suppose, from this table is that through the project, we've managed to come up with a couple of different modes of action for most of the crop groups, and that gives us a bit of ability to rotate chemicals and stuff like that going forwards. And it also means that if you happen to get a resistant population come in, we might have another option already. And I'll also note that through the project, we've looked at developing some permits for some additional chemicals. At the moment, we haven't gone forwards with things like neem and cyanotranoprol, which appear to be fairly effective. We've got data that would support a use pattern and that sort of thing. But there's a fair bit of work in just keeping these permits going over time. So at the moment, we've sort of prepared them, put them on the shelf, and the idea is that if Leafminer comes further south, then we'll be able to enact them. And just one more thing is that, again, with all of these chemicals have translaminar effects. So it means they can penetrate and get into the larvae, and they also have fairly low toxicity towards most of the beneficials, and that fits in with your IPM stuff. So just a quick summary, I suppose. So when selecting any sort of chemical, we want to use it only when really absolutely necessary. So once it actually gets into production areas, we'll need to develop some of those threshold, economic thresholds and stuff about when to actually control it and when to allow the beneficials to do their own thing. And that the conservation of the beneficials is definitely an essential part of IPM and managing this pest. When using chemicals, we want to, minimise disruption to the parasitoids. Preferentially, would select for systemic and translaminar chemicals if we're looking at control options. And we also want to sort of consider resistance going forwards as well, just to try and make sure we've got some different options available. So through the project, we've managed to develop permits covering 5 pesticides in the vegetable, nursery, melon, potato and onion industries. So yeah, that's just, chemical control in a nutshell, I suppose. So thank you.
All right, so we have a question from Dayton. In South Australia, we grow most veg seedlings and flowers. Never see them in any veg seedlings, but always find mines during September through to December, only in dianthus. Why are they so attracted to the dianthus and not in brassicas, peas, beans and spinach?
Most of the agromyza species that we have in the world have a fairly narrow host range. I suspect that the most likely species that you're looking at in South Australia, it would appear to be Leafminer Paris, although I'm surprised that it doesn't attack spinach, but certainly that's the timing that you're talking about is about right for that species. It would be well worthwhile getting a sample taken just to confirm, but I think that would be a good idea, but I imagine it's highly likely to be Leafminer Paris, and it does have a very specific host range, and it certainly you wouldn't expect it to go into brassica. So that would be my suggestion with that particular one.
Thanks, Peter.